calcifying epithelial odontogenic cyst makes cystic cyst, cystic lesion in the odontogenic cyst, which is having the synonyms like calcifying odontogenic cyst, keratinizing, and calcifying odontogenic cyst, calcifying cystic odontogenic tumor. Gorlin said, Gorlin histological resemblance is the calcifying epithelioma of mouth, not actually a cyst but a neoplasm. So, incidence is less than 1%. Age predilection indicate gender predilection is equal. Side predilection more in maxilla, premolar, and canine region. Clinical feature asymptomatic swelling may or may not be present. Lingual expansion may sometimes be observed. Displacement of adjacent teeth, extraosseous lesions, pink to red. Radiolucent, radiological features of calcific cyst is as a unilocular or multilocular radiolucency. Outline is well demarcated, may or may not be regular one. Radiopacities are present. So radiolucent as well as radiopacities are present. Remember this thing. Displacement of teeth occurs, resorption of the teeth occurs, and extraosseous lesions can be seen. So the lining, epithelial lining is present. Prominent basal cell layer is palisaded one. Budding from basal layer, the epithelial proliferation into the lumen is seen. Tubular dentinoid juxtaepithelial material is seen underlying the epithelial lining. So histopathologically, you can see the epithelial lining in which you can see the amyloblast-like cells. Remember the word amyloblast-like cells, in which they are the tall columnar cells are present. Their nuclei is palisaded one. Then it is having also the striate reticulum-like cells, which proliferate into the lumen. So uh, lumen is having striate reticulum-like cells, and epithelial lining contains the single amyloblast-like cells. Underlying the epithelial lining, there is a tubular dentinoid or juxtaepithelial material is there, or dentinoid-like material is seen, and after that, the capsule. That means the fibrous wall. More important characteristic feature which can be seen in the calcified odontogenic cyst is the ghost cells. These ghost cells are enlarged, balloon, ovoid, or elongated elliptoid epithelial cells. It is having the outline well defined, only nuclear remnants of faint outline of original nucleus can be seen. It is having tendency to calcify. So ghost cells are pale eosinophilic swollen epithelial cells that have lost the nucleus but show faint outline of the cellular and nuclear membrane. They contain many chromofibrils and cytoplasmic organelles. The calcified odontogenic cyst is often encountered in association with an odontoma. Sometimes amorphous eosinophilic material may be identified in juxtaposition to the proliferating lining epithelium or intermixed with the ghost cyst, and this is called as the dentinoid material. When the material is formed in the abdominals, and the lesion may be termed as a dentidogenic ghost cell tumor. The presence of ghost cells within the proliferating odontogenic epithelium, the presence of ghost cell keratinization is not sufficient enough for the diagnosis for, and nor it is the pathognomic one. So it is a characteristic feature of the calcified odontogenic cyst, but not the diagnostic feature. It is not a diagnostic criteria. So SAT question can be asked on ghost cells separately. So ghost cell keratinization is observed in other than this cyst is the odontoma, amyloblastoma, amyloblastic fibrodontoma, amyloblastic odontoma. And these are characterized by loss of nuclei, clear preservation of cellular outline, resistance to resorption, induction of foreign body granuloma, and the potential to calcify. So ghost cells differ from the normal keratinocyte squamous cells In that they are larger one, often vacuolated cells. Remnants of nuclear membrane are more prominent. Ultrastructurally show stonofilaments and hydroxyapatite crystals. So treatment is the surgical enucleation. If it is present with any benign neoplasm, should be treated like a neoplasm. Next important cystic lesion is the glandular odontogenic cyst. Synonyms are the odontogenic cyst, mucoepidermal odontogenic cyst. So glandular odontogenic cyst, a cyst arising in the tooth-bearing areas of the jaws and characterized by an epithelial lining with cuboidal or columnar cells, both at the surface and lining threads or cyst-like spaces within the thickness of the epithelium. So it is drawn attention due to high recurrence rate and aggressive growth potential. So it's very similar to your odontogenic keratocyst. It is having the high recurrence rate and the aggressive nature. Incidence is less than 1%. Age predilection 6 decade. Gender predilection male. Side predilection anterior mandible, premolar, and canine region. Very important one. Side predilection anterior mandible, as well as premolar and the canine region. All the cysts are occurred in a posterior region. Only COC and GOC are occurring in the anterior region. Asymptomatic swelling may be present or not present. Displacement of tooth occurs. So radiographical GOC represents as a unilocular or more commonly a multilocular radius. The margins of the radius are usually well defined with a sclerotic ring. Remember the sclerotic ring was present in a OKC also.
Hmm.
The cyst is lined by squamous epithelium. The epithelial cells may be cuboidal or to columnar and have an irregular and sometimes the papillary surface. The mucous cells may or may not be present within the epithelium. In focal areas, the epithelial lining cells may form spherical nodules similar to those seen in a lateral periodontal. The fibrous wall is usually devoid of any inflammatory cell infiltrate. So its lining is combined with squamous cells. These squamous cells are cuboidal to columnar one and are having irregular and sometimes the papillary surfaces. The focal areas of epithelial lining may show spherical nodules and sometimes they are very similar to your lateral periodontal. We are going to see the what is lateral periodontal afterwards. So columnar cells with cilia, stratified squamous cells, small microcyst of the clusters of mucous cells. So five major criteria: squamous epithelium, varying thickness, cuboidal eosinophilic cells, mucus cells, or you can call it as a goblet cells, which are present in a glandular odontogenic cyst. That's why the name is given as a glandular odontogenic cyst, and intraepithelial glandular or duct-like structures. Next important cyst is the gingival cyst of adults, which is a developmental odontogenic cyst of the gingival soft tissue derived from the rest of the dental lamina, contain a lining of embryonic epithelium of cuboidal cells. So, two recognizable forms of gingival cyst arising from cystic transformation of dental lamina or rest of serous, and second recognizing criteria is arising from traumatic implantation of the surface epithelium. So, Viscosis et al. It concluded that it does arise from the post-functional rest of dental lamina and thus represents the extraosseous counterpart of the lateral periodontal system. So, clinical features more common in adults over 40 years of age. Site is canine premolar area, small, well circumscribed lesion on the gingiva, usually seen in the attached gingiva or interdental papilla. The gingival cyst of adults, you just remember the cysts which are occurring at anterior region. First was the COC, second was the GOC, and third one is the gingival cyst of adults. So histopathologically, epithelium ranges in thickness from 1 to 3 layers of the flat cuboidal cells to a rather thick stratified squamous epithelium without the radiages. So many epithelial cells have pyknotic nuclei and show perinuclear cytoplasmic granularizations. So you can see the stratified squamous epithelium and underlying connective tissue stroma or the fibrous wall. And here the white part is the cystic lumen. So glycogen-rich clear cells are present especially in the focal thickening of the lining. Rest of dental lamina may be present in the connective tissue, may or may not exhibit an associated inflammatory reaction. Next important cyst is the gingival cyst of infants. It is also multiple or solitary superficially raised nodules on the edentulous ridges of the infant. That means the newborns can undergo this type of gingival cyst derived from the rest of dental lamina. So Bohn's nodules or extens pearls are two similar lesions with this type of cyst. The extens pearls are the keratin filled nodules found along the midline raphe, probably derived from the entrapped epithelial remnant along the line of fusion, and Bohn's nodules are the keratin filled cysts scattered over the palate and apparently derived from palatal salivary gland structures. So Bohn's nodules and extens pearls are very similar structures, but they will be distinguished because of their formation and the location. Clinical feature: small, discrete, white swelling on the alveolar ridges, asymptomatic and produce no discomfort to the infant. So true cyst within thin, histopathologically they are the true cyst within which the epithelial lining which lack the retiridges, so there is the no retiridges of the epithelial lining. Lumen is usually filled with this squamated keratin, occasionally containing the inflammatory cells. Next important cyst is the lateral periodontal cyst. It occurs on a lateral periodontal position. In order to diagnose it, an exclusion of the inflammatory origin and the possible OKC must be ruled out clinically and histologically. It develops as a dentigerous cyst along the lateral surface of the crown, and as the tooth erupts, the cyst assumes a position in approximation to the lateral surface of the root. So you can see in the picture, diagram A represent a tooth structure in that a laterally occurring cyst at a lateral surface. Here is the epithelial lining, and when this tooth erupts in the oral cavity through the gingival region, then this cyst goes lateral. So lateral dentigerous cyst can convert into a lateral periodontal. So this is the first theory. Second theory, proliferation of the rest of malacis as a primordial cyst of the supernumerary teeth, more occurrence of a supernumerary teeth in premolar area than proliferation of cyst of dental lamina. Scorsese et al. Concluded that the lateral periodontal cyst and gingival cyst of adults share a common histogenesis from the post-functional dental lamina rest, and that these two cysts are basically central, intraosseous and peripheral or extraosseous manifestations of the same region. So clinical feature chiefly in adults, predilection for canine premolar area, and also male and female is equally hampered. Radiographic features, you can see clear cut. It is present lateral to the both the tooth, premolar and the canine. Here is the canine, here is the premolar. So histopathologically thin, non-keratinized epithelium, usually 1 to 5 cell thick, which resembles reduced enamel epithelium. That means the finding is very similar to your dentigerous cyst. Cuboidal or columnar cells may be seen in the lining epithelium. So focal thickened plaques of proliferation lining cells may be seen projecting into the lumen. Many of the epithelial linings have a clear vacuolated glycogen-rich cytoplasm. So this cyst is very similar histopathologically to your Dentigerous cyst, in that it is formed because of the radiosynomal epithelium cells. That's why the 2 to 3 layer cell layer thick tubular cell lining is there, but the history is different. That is clinical finding. In dentigerous cyst, you can see the impacted or embedded tooth, whereas in lateral periodontal cyst, there is the history of eruption of the tooth. Next is the botryoid odontogenic cyst. This is the cystic lesion with clinical and radiological features of a lateral periodontal cyst. So Withers and Waldron first suggested the term botryoid odontogenic cyst because the gross specimen resembles a cluster of grapes. So in lateral periodontal cyst, you can see a single lumen, but in case of botryoid odontogenic cyst, there are many lumens present in a single cyst. And that's the main difference between these two. So the name of the botryoid odontogenic cyst reflects the gross similarity to that of the clusters of grapes. Origin of the pathogenesis. So cells develop a single cyst and forms a lateral periodontal cyst, and cells develop in a multiple luminal cystic lesion and form the botryoid odontogenic cyst. There is a feature 5 to 7 decade of life, more in the anterior mandibular region and usually asymptomatic but may cause pain. Mostly it is multilocular radiography but can be unilocular as well. Histopathologically, similar to lateral periodontal cyst, it is a multicystic with thin fibrous connective tissue septum. Cystic cavities are lined by a thin non-cranial epithelium but occasionally may be lined by stratified stromas. Lining may contain flap-like epithelial layers, very similar to your lateral periodontal cyst. Clear cells are also present. Next is the inflammatory cyst. So we completed the odontogenic cyst. Now inflammatory cyst or periapical or a radicular cyst. Synonyms for the radicular cyst: apical periodontal cyst, root end cyst. It is not most common cyst. So etiology of the periapical cyst: it is an infected tooth, or whenever the infected tooth occurs, it remains untreated. Then the infection goes to the pulp and toxins to the root of it. So it causes a periapical inflammation, which stimulates the epithelial rest of molasses found in the apical periodontal ligament and forms the periapical granuloma. And these granula or epithelium cells undergoes the necrosis caused by lack of blood supply and forms the cyst or the lumen. So sequelae of pulpitis causes the sequelae of pulpitis, pulp necrosis, apical periodontitis, and the periapical cyst. So first thing, when your infection occurs, it goes to the dentine, enamel, dentine caries occurs, enamel, dentine, and causes to the pulp. It goes periapically and forms the pulp necrosis and causes the apical periodontitis and the periapical cyst. The next formation, the periapical granuloma and the cyst. So like this, the cystic lesion. The white line, this black one is the lumen, whereas the white is this is the epithelial lining and the epithelial capsule. So caries, tooth fracture, improper restorations causes the pulp necrosis, causes the periapical granuloma. So this is the pathogenesis of periapical cyst. Pathogenesis: granuloma is highly vascularized tissue containing a profuse infiltration of immunological component cells, that is lymphocyte, macrophages, and the plasma cells. The epithelial rest of Malassez pluripotent cells can differentiate into any type of epithelial cells under proper stimuli. So periapical granuloma causes the rich blood supply, rest of cell Malassez proliferate, and mast cells of cells. The continuous growth deprivation of the nourishment, inner cells of the mass undergo liquefaction necrosis and causes the leads the formation of cavity which is located at the center of the granuloma and give rise a radicular cyst. Source of infection. Source of epithelium are the rest of molluscs, maxillary antral lining, sinus tract epithelium, presence of pre-existing granuloma or de novo, cyst formation three phases: phase of initiation, phase of cavity formation, and the phase of cyst enlargement. Phase of formation. Cyst cavity forms within a proliferating epithelial mass in an apical granuloma of degeneration and the death of cells in the center. The proliferating epithelial masses show considerable intercellular edema. These coalesce to form microcysts containing the epithelial and the inflammatory cells. Some products of dead pulp initiates the process, same time evokes the inflammatory reaction. So in phase of initiation, the proliferation of epithelium is associated with the presence of active inflammatory cell infiltrate and polymorphonuclear leukocytes. So local changes in the supporting connective tissue may be responsible for activating the cell nest and decreases the O2 and increase the CO2 tension, and local reduction in pH produced in chronic inflammation may be critically factors. So mechanism of cyst development. Cytokines causes the proliferation of the epithelial cells. So these cytokines are responsible for the central cell breakdown, early lumen formation. Then, because of the H2O tension, cyst expand because of the osmotic gradients, and finally the formation. Cyst maturation matures with flattened surface cells, a basement membrane, and a connective tissue cell wall. Osmotic gradient continues, cyst lining proliferates, and cyst expands. So cyst development begins and continues by cytokine stimulation of epithelial rest and is added by the central cellular breakdown products which create a hyperluminal solution that transmutes fluid for further cyst expansion. Phase of enlargement raises the osmotic pressure of the cyst fluid, increase polyaginolytic activity, prostaglandin induced bone resorption. Really features 60% of all jaw cysts are radicular cysts. All types of dentition, deciduous dentition seldom seen. Peak incidence is third to fourth decade. Males are more commonly affected than the females. Rarely grows to size where it destroys the bone. Rarely produce expansion of the cortical plates. Radicular cyst is a lesion that represent a chronic inflammatory process over a prolonged period of time, may be asymptomatic. Radiographic features: round to oval radiolucency of variable size, generally delineated, mostly having radiopaque, cyst or granuloma, lesions larger than 2 cm in diameter, rarely induce the root resorption of the affected tooth. Radiological findings of the periapical cyst: well-defined radiolucency with sclerotic borders. Histopathological features: epithelial lining, non-keratinized stratified squamous of variable thickness and sometimes the incomplete one. Epithelial lining can be discontinuous due to areas of intense inflammation. According inflammation, arcading pattern can be seen. That means arcading patterns of the rete ridges can be seen. That means in the epithelial lining there is a entrapment of the connective tissue and it is called as the arcading pattern. It is a very important epithelial lining feature of the radicular cyst. So keep actively proliferating irregular hyperplastic type if infected, ridges, arcades, snake-like appearance. Lacks a defined basal cell layer, ciliated cells with maxillary cyst. Histopathologically, cystic lumen, epithelial lining of forking and arcading ridges, connective tissue capsule. Hyaline bodies or Rushton bodies can be seen in the epithelial lining of the radicular cyst. So, what are the hyaline or Rushton bodies? Great numbers of amorphous in structure, eosinophilic in reaction, and brittle in nature. So, these are the Rushton bodies. Elliptical, oval, or tubular in structure. All these are the eosinophilic hyaline bodies or Rushton bodies. So, lumen of the cyst contains fluid with low concentration of protein that stings paleoeosinophilic, containing a great deal of cholesterol, rarely limited amount of keratin. So, cholesterol clefts are seen in a connective tissue capsule. So, cholesterol released from the disintegrated RBCs and from the serum accumulates in the tissue because of its inaccessibility of normal lymphatic drainage. Close relation between the occurrence of cholesterol and hemosiderin containing macrophages as well as free hemosiderin in the tissues. These are considerable accumulation of cholesterol could occur through degeneration and disintegration of lymphocyte, plasma cells and macrophages taking part in the inflammatory process with the consequent release of cholesterol from their walls. Once the cholesterol crystals have been deposited in the fibrous capsule, they behave as a foreign body and excite foreign body giant cell reaction. So giant cells are derived pericytes of the vessel wall. So sometimes you can see the giant cells also with association with the cholesterol clefts. So complications, epidermoid carcinoma and mucoepidermoid carcinoma. Extraction of the involved tooth, curettage of the periapical tissue, root canal treatment with apicectomy of the cystic lesion. So this completes your odontogenic cyst chapter. So this is the wall. This is the picture of OKC. You can see here the keratin, abundance of the keratin in the cystic lumen. These are the lining, corrugated lining of the epithelial surface. See the corrugations present on the lining, and these are the pycnotic nuclei suggesting the parakeratinized lining. Then it is having 6 to 10 cell layer thick, and the basal cells are showing palisading lining and underlying connective tissue capsule is seen. Next picture is the dentated cyst. It is having the reduced enamel epithelial lining, 2 to 3 cell layer thick, and underlying connective tissue capsule. Sometimes it may contain the mucous cells and the fat cells.